All right, let's get started. Thank you for joining us today for another round in our webinar series on investigations of biomedical issues and how they relate to human disease. First, I'd want to thank Gary Watkins, Director of Marketing at AnaBios, for putting on this webinar series. So if we can switch to the next slide. So first of all, I'm just going to give a quick intro. AnaBios is a unique CRO organization and biotech company that is based in San Diego. We recover human organs from ethically consented donors and use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first in human, and working with AnaBios is as close as you can get to a clinical trial without doing a clinical trial. For those doing pulmonary research, in the past year, we've introduced the availability of human precision cut lung slices. And actually, we have had a webinar in the past, which you are free to listen to. And this slide shows the talk by doctors Ramaswamy Krishnan and Joseph Brain from Harvard Medical School and Public Health on the usage of precision cut lung slices in their research. The talk is recorded at the AnaBios website, as I mentioned. If we can move to the next slide. So we recover human lung on a regular basis. We create slices which are cryopreserved and shipped to customers. Their cryopreserved slices can be thawed and placed in culture, remaining viable for days. Typical slices are shown at the bottom. You can download the brochure on PCLS from our website or directly from this webinar. If you note under handouts, the brochure is listed and is downloadable. Okay, for the next slide. So without any further ado, it's my great pleasure to introduce today's speaker. Dr. Scott MacDonald obtained his PhD in cardiovascular physiology at Temple University in Philadelphia. Following that, he completed a postdoc fellowship at Temple in the lab of Steve Hauser, where he focused on identifying mechanisms responsible for the pathogenesis of heart failure. His work, recognized as best paper in Circ Research in 2010, examined the role of CAM kinase 2 in altered contractility, myocyte apoptosis, and transcriptional regulation associated with heart failure progression. Scott has been recognized by the International Society for Heart Research and awarded the Young Investigator of the Year. After academia, Scott spent 8 years as a principal scientist at Boehringer Ingelheim, working on novel therapeutic treatment options for chronic kidney disease, heart failure, and fibrosis. Currently, Scott is Associate Director at Regeneron Pharmaceuticals in the Department of Cardiovascular and Renal Disease, where he leads an in vitro and histology group supporting both screening and functional assay development for heart failure, systemic hypertension, and pulmonary fibrosis. And the title of his talk today is Using Single-Cell Sequencing to Identify Distinct Cell Populations from Fibrotic Tissue. Scott, please take it away.
Yep, thank you for that kind introduction. And hopefully I would now and we'll have a good consistent webinar here. But I do want to thank Richard and Gary and AnaBios for the invitation to share some of our work. Really excited to be able to present and share some work we're doing to actually apply single-cell sequencing to identify distinct cell populations from the fibrotic lungs. And I think that's sort of a nice connection to the precision cut lung slices that we are using in our lab as a translational model. So we're kind of taking a step back here, and I'll show you some of our mouse work, but we do see great opportunity for the lung slices as well to translate some of the findings that I'll share with you from our culture and animal models. see here. All right. So just as a very quick agenda, so I hope the slide deck, I've truncated it a bit so that I can really move through some key points, make some key observations, share some insights, with the goal really of leaving time at the end of the hour to have a robust discussion. And I look forward to any questions, feedback that anyone in the audience may have. So just going to provide then a really brief intro on fibrosis and different cell types and some of the work that's been done, at least from my perspective. And while, of course, the utility of single cell sequencing to explore cell type distribution from different organs, different tissues, is really, gaining a lot of traction over the last few years. I do want to actually start with just some basic observations that we used exploring single cell sequencing in culture-based systems. And kind of how that highlighted its utility, and its potential, insight that this type of an approach can provide, but also highlight some of the the challenges and some of the things we need to take into consideration when interpreting the data. And then I'll summarize and hopefully open for questions. So when we talk about fibrosis, it's a very wide and big term. I kind of look at it from a very high level as it's wound healing that doesn't go right. It's wound healing where there's no defined edge, no defined edges of the wound, and really, it's something that is really impactful for human health because it's associated with a number of different organ-based, diseases, so a number of different conditions. And of course, fibrosis is also associated with aging. So in essence, it impacts all of us. But really what it comes down to is you have extra or excess extracellular matrix, so fibrotic tissue, connective tissue, that disrupts the normal architecture of your tissue, leading to alterations in function, increases in stiffness, in pressures, and disruption, you said, of the normal function over time. And this process, leads to about 45% of deaths, or again, because it impacts all of us and it's across organ systems, many deaths are associated with some kind of chronic fibroproliferative disease, basically meaning when they look in the tissues, there's some type of extracellular matrix and fibrotic deposition that's associated with the outcome for that patient, or unfortunately the death of that patient. Now, despite these facts, there really are very few treatment options that are directly targeting the development, the progression of this extracellular matrix of the fibrosis. If you look at the field right now, there are many things in the clinic and again, significant preclinical work to explore pathways and ways to intervene in this detrimental process. But there are only a few drugs that are actually approved and have shown definitively that they impact the development of fibrosis, and primarily in the lung. So I'm going to focus the talk and our data on our lung experiments. But nintedanib, which is a kinase inhibitor, has shown benefit in IPF and now scleroderma with ILD, and pirfenidone, which is anti-inflammatory, anti-TGF, really mechanistically a little bit unclear as to what it targets, but it has also shown benefit in lung fibrosis. So despite these staggering statistics and the impact across multiple organ systems, there's still a really high unmet need for novel therapies that will address the fibrotic development. So where to start? Lots of different places in fibrosis work, and one of the places that I've done a lot of work over the years was trying to understand, and not just me, but the field in general, I'll say 20 plus years here, where there's been work to try to understand the cells that are responsible for the development of fibrosis, or the cells that give rise to the fibroblasts or the myofibroblasts, the disease-associated fibroblasts, sometimes called myofibroblasts. And this work is focused on identifying, again, specific cell types that may differentiate into blast-like cell, like fibrocytes, endothelial cells through endo-MT, epithelial cells through the process of EMT, epithelial to mesenchymal transition, either quiescent fibroblast just becoming activated and kind of moving from a more quiescent state to, again, what's termed a myofibroblast or an activated fibroblast, and pericytes that may sort of dislodge from their vascular support role and migrate interstitially to become a matrix producing, a collagen producing cell. And again, this, if you look at sort of the problem, it's twofold. One is we can work to understand the cells that give rise to the disease fibroblast, or if we take maybe not a more simple approach, but a more basic approach and kind of thinking, can we first identify that there is truly a disease-specific population of fibroblasts? And if so, can we distinguish those from your quiescent, healthy, necessary fibroblasts? And can we target these cells specifically? And coming now from Regeneron, we're an antibody company, so we are looking for ways to deliver therapies, not just targeting circulating factors, but targeting specific cell types. So one of our goals would be, is there a cell surface receptor on an activated fibroblast, a disease-associated fibroblast that we can actually target to deliver, an inhibitory antibody or potentially a small molecule as an antibody drug conjugated tool? Are there internalizing receptors on these disease-associated cells that we can take advantage of? Therefore, we limit and we protect the organ from the excess fibrosis without impacting the normal wound healing process that all of these cells play a role in. And that's where we started. And as I mentioned, where did I start? So this is probably going back now many years, back to the basic observation that I continue to see whenever I cultured fibroblasts in a dish. And actually just wanted to have a caveat here that these cells are being grown on plastic. And I know in the fibrosis field that is a challenge, because the plastic as a stiff matrix will induce activation of the cells and can change the phenotype. So that's understood here, but in essence, they are cultured on plastic. And these are normal human lung fibroblasts. So what I really appreciated in many years of putting cells into culture like this, I really always saw and visualized and had this observation that there are distinct populations of cells even within culture. And these populations, so what we're staining for here in the green is smooth muscle actin, and in the red is collagen 1. And really what we're seeing is the intracellular pro-collagens, obviously inside the cell, and the blue is DAPI. So you see a really nice confluent monolayer, and you see cells that are primarily or exclusively, in this case, expressing collagen, and you see some cells that are primarily expressing smooth muscle actin, although when you do look closer, you can see that those same cells are also expressing collagen. So they're all fibroblasts, but there are distinct populations that are even evident in culture. And just to be, really upfront, what we used to do is obviously take these plates, we would, homogenize and put trisol and collect them and really sort of mix everything together and look at the gene expression patterns as a whole, with really, not having the ability to sort of understand what's unique about these smooth muscle expressing cells, which again, some of us might call myofibroblasts, contractile, and what's unique about these cells that are not expressing the smooth muscle actin. And we lose that when we do bulk RNA sequencing. So we ran a very simple in vitro experiment, again, really low power, but it was just proof of concept with this single cell technology. And we did this a number of years ago as we were learning and sort of appreciating this new technology to sort of look at these cells and understand the gene expression pattern within these cells, not as a whole, but as unique. Each cell is a unique entity. So we took these healthy lung fibroblasts and just two different commercially available donors that we had that had IPF, so a diseased and healthy lung fibroblasts. We put them into culture. We treated them, let them grow to about 80% confluence. We serum starved them overnight, and then we treated them with TGF-beta for 24 hours. The 24 hours is acute, I recognize that, and I think you could definitely see different patterns of gene expression if you were to target and treat these cells for shorter or longer period of times, but for this data set that I'll share, we looked at a 24-hour point. We take take these cells, lift them up off the plate and trypsinize them and move them through, we just call 10x, but it's 10x genomic technology. So this is the machine on the left. And basically this is a really, amazing and efficient way of characterizing each individual cell. So what the machine will do is with your single cell suspension, and whether that's coming from your organ of interest or in this case a culture plate, it takes those cells and it combines them and it tags them with unique barcodes, and it encapsulates each of those cells with the unique barcode within a lipid droplet. The reverse transcription occurs within each of the droplets, and then the sequencing libraries are finished in a bulk reaction. What this allows you to then do is deconvolute. So once you have the bulk reaction and the barcoded RNA, you can deconvolute and actually go backwards and determine which cells the RNA was derived from. So you get gene expression per cell. It's fairly quick and efficient. We can run 8 samples per run. It only takes about 10 minutes. So you can run multiple samples as you have, if you have more than 8, pretty efficiently. So just to show some of the QC, and when we get into the in vivo data, I'm not going to have a QC slide, but I just wanted to show, because again, this was a proof of concept that we were running to understand the technology, what we could gain and what we may lose. So when we apply sort of a threshold of QC, what we're looking for is that each of the cells, so each of the cells is a different dot, and the different groups are either the IPF patient 1 plus or minus TGF beta, IPF 2 plus or minus TGF beta, and then we're calling the normal human lung, which are the far right in each of the figures here. And what we said is kind of a general threshold. And when I appreciate and read manuscripts, it's always good to see these QC parameters because they do differ depending on groups. But we apply that each cell needs to have at least more than 200 genes in order to make it through to the analysis. The unique molecular identifiers, which is the tags, the barcodes, greater than 0 per cell, and we're well above that. And then fraction of mitochondria. So again, looking at mitochondrial reads as an index of cell viability and health, we like to keep those less than 20%, and you can see we have really good consistency here. Of course, the QC coming from tissues can be a little bit more challenging because of the isolation process and the timing, where it's coming from a culture condition like this is usually going to be a little bit cleaner. And you have a more homogeneous population of cells to start with. So all in all, we sequenced from these 3 different donors, we sequenced about 9,000 or 8,700, just over 8,700 cells, and we took the analysis and we plotted them using a Tisney algorithm. So this is a spatial distribution of the cells based on the unique and differential gene expression of each cell. So each of these dots is one cell. So again, about 9,000, a little under 9,000 total dots on the plot. And each of these groups are grouped together based on the similarity, the similar gene expression patterns and the distance. So further groups, the distance between, it shows that there's greater or a more distinct gene expression pattern of these different groups. So the first thing to note, and again, this visualizes it, I think, quite nicely, is that the human lung, which is the blue, they cluster in a distinct, they form a distinct cluster, I should say better, versus the 2 IPF donors, which are the red and green in the upper left. One important sort of first take home that I want to share, and again, I had seen this many times, and sort of, again, my belief in TGF beta is that TGF beta, you can see here, the normal human lung fibroblasts, when you treat them with TGF beta, in this case for 24 hours, you do regulate thousands of genes. The matrix genes are turned up, contractile proteins, genes are expressed, and their proteins, are generated. So you can see that the TGF beta induces a differential gene expression pattern from just the normal serum-free condition, which was in the blue. And we also see that in the IPF. So even though the IPF cells are, in essence, distinct from the healthy controls, TGF beta still does induce a robust gene expression pattern, and we know that. TGF beta is the one we all use in fibrosis to sort of activate cells, with the idea that applying TGF beta to a cell induces a diseased pattern. Applying TGF beta, maybe to say it in a better way, applying TGF beta in culture to a cell like these human lung fibroblasts induces a myofibroblast, a disease-associated cell. And sort of the first point here is I don't believe that. What I see right here is that the normal human lung fibroblasts, when treated with TGF-beta, they actually form their own unique cluster. They actually don't overlap with the IPF or even the IPF treated with TGF-beta. So yes, I'm not putting down TGF-beta as a stimulus to activate fibroblasts. It does, and it can be used, but it shouldn't be used in a way, at least at this 24-hour time point, to induce a disease-associated cell, because again, the patterns don't overlap. So it is creating an activated cell, but not necessarily, in this case, an IPF, a cell that's relevant to the IPF condition as much as we may have assumed in the past. And I just wanted to note one other observation. Again, this is coming from the IPF clusters, and I think something you may have noticed on the last slide was that the IPF clusters, independent of TGF-beta, there were two. So there's an IPF, we called it cluster 2, and an IPF, we called it cluster 4. And these two clusters here are fairly unique based on their spatial distribution here. But when you look at the genes expressed in either of these, it kind of comes back to that initial observation that I had with the healthy cells. In the plate, there are distinct populations, and we can learn about those distinct populations by applying single cell. So proof of concept, at least from a dish, is we can find cells that have, in this cluster 2, for instance, high expression of matrix genes. So the collagens, fibronectin, MMPs, so sort of genes associated with matrix production or degradation, so turnover. When you look down into cluster 4, the genes that were primarily expressed there, and this was greater than 5-fold difference over the other cluster, were contractile proteins, genes for contractile proteins. So it really kind of makes the point that single cell, even from a controlled scenario like culture, can pick up distinct populations. And we are seeing cells that are contractile. Again, they're still fibroblasts, they express collagens, but they are more contractile, and we do see cells that are more matrix producing cells. So based on that initial proof of concept, just being able to sort of peel apart cells in culture and see that we can use single cell sequencing to identify different populations, we then applied this technique to our lung fibrosis model. And the model that I'll share is one that's very commonly used. We use a mouse bleomycin model. We do an aspiration-based model. So the animals, the small bolus of bleomycin is actually added right above the trachea, and they sort of inhale and aspirate it down into the lungs as a model, so not direct tracheal instillation. We have done bulk gene expression on lungs, and you can see that in this model you have really rich and robust upregulation of matrix genes, in this case just collagen 1A1. And on the bottom right, this is taking total collagen. So we induced the fibrosis with bleomycin, and we took animals down on day 3, 7, 10, 14, 21, 28, and out to 35. And you can see this is just total collagen micrograms for the total right lung. And you can see a really nice increase that does lag a bit behind the gene expression, which makes sense. The genes come up quite early as an initial response to the insult, and then the matrix starts to get laid down, and you can see statistically significant increases by day 14 that starts to progress. In the model, out past day 35, you can start to see it plateau, and even you do start to see some resolution there. So we normally take our animals out to day 21, where we have a nice window for both gene expression changes still and the matrix deposition. Sorry, for the single cell sequencing part, we were really challenged to run an initial study on the day of interest. And what is that day of interest? I know for anyone that does in vivo models, it's quite challenging to say, we always want to look across a wide range of time points. And that can get quite challenging, especially when we're isolating and doing single cell sequencing. So for this study that I'll share, we actually selected day 11. This is early in the process. It's really kind of the first time where we're starting to see between day 10 and 14 an increase in the actual matrix production. We know it's still, within a point of where the initial insult induces a rich inflammatory response, but it's really at that upstroke. So it's at the start of the fibrosis. We're really interested in kind of learning a bit more about what's happening at these early time points. But I fully admit having additional early plus later time points would be and will be an important aspect. And there's been some nice publications over the last year where full time course data have been generated and are available in the public domain. So really nice work that's followed up on some of these experiments and really built out great data sets for the field. Not to spend too much time here, these are just our methods that we use to do the isolations. And I'm just kind of pointing here with my laser pointer there. One thing that we find really valuable in doing cell isolations from the lung is actually doing tracheal installation of our enzyme cocktail, our protease solution. We get it right down into the lungs, and then we're able to isolate, remove those lungs, and further process them once they're outside the animal. But we really have good, consistent 90+% viable cells, and we're able to move those through to the lung, to the 10x platform. And this is really an important point in optimizing the process and the timing, because really for 10x and for single cell sequencing in general, the cells that go in, your ability to fully, as best we can know, fully digest, capture all the unique cell types, and move those quickly to the 10x platform so that you're able to at least capture as many distinct cell populations as possible. You really need to optimize the digestion and the collection and the timing for your individual models and systems. So for here, we had 3 different groups in the study. Saline, the bleomycin, so our injured model, our fibrotic model. We had N's of 3 to 4 per group for this study. We also did BID dosing of nintedanib at 60 mg per kg, which is the usual dose, or a dose that we've shown benefit in our model system, as well published in the mouse to show benefit. And of course, this is I think a really important aspect of the study, in that this is standard of care, really one of, I said, only 2 drugs that's approved in lung fibrosis. So we work this into the model, and we analyzed just over 18,000 total cells across the groups. I'm sorry. So when we take those cells and we apply the same TISNE algorithm, now we have 20,000 cells instead of the 8,000. We are able to see that we do observe about 17 distinct clusters. And what we can do is take the gene expression patterns of each of those individual clusters and align those with gene expression, known gene expression patterns for individual cell types. And then we can label each individual cluster defined by its gene expression and known genes expressed in those cell types by each cell type. And here you can see it's several large clusters, macrophages, smaller vascular smooth muscle cell cluster. But you do see on the right here in this peach color, a fibroblast cluster that's fairly robust as a percentage of the total population. And it's great to be able to see it like this, where you can see and label each individual cell type. I think that's always the first step for single cell sequencing, and TISNE, and some people are now using UMAP algorithms, which gives you a very similar output. But then we can overlay the different groups, and also important to recognize and look for different patterns to make sure that each of your groups, that cells from each of your groups are represented in your main populations. And just pointing here at the fibroblast population, blue, there's overlap. So we were able to fibroblasts from each of the different groups in the study. Now, you might have noticed as I sort of glanced over in that last slide, looking at the different groups, were changes in the different group populations. It's a little hard to see that when you have everything clustered together. So what we did is we pulled out just the cells that were derived from saline animals and just those that were derived from the bleo. So just these 2 groups, so non-overlapping. And what I think you can appreciate here is that in general, there really is good overlap between the 2 populations, or between the 2 plots here, where most of these groups have cells derived from both the bleo and the saline. But I definitely am highlighting here, sorry about the phone. What I'm highlighting here is the macrophage population. And the macrophage population is interesting because we took this study down on day 11, and I noted day 11 is where we start to see matrix deposition, but we do see inflammation, lots of inflammatory cells infiltrating the lung. And I think these are key aspects and key drivers actually of the disease progression. And you can see in the bleo there, so most of the macrophages actually are derived from our disease population. And these are just the averages then. Each of these bars is one of the individual animals, and these are the different cells, the different populations, percentage of cells that were derived from each of those populations. And I think what you can appreciate here is that the macrophage population, 9% of the total macrophages, the total cells captured from the saline were macrophages, and about 30% from our bleo and also our nintedanib treated group. One thing that from the fibrosis angle was this population. And as I mentioned, there is a nice representation from each of the different groups in this cluster here, this fibroblast cluster, which is here. And one sort of interesting first take home was that we actually didn't see any changes in fibroblast number between the groups. You know, I guess I would have said walking in that you're going to have more fibroblasts coming from bleo. Now, this may be, you know, a caveat of the isolation process by which you're not able to isolate all the interstitial fibroblasts from those matrix-rich areas. So again, that's a limitation potentially of the digestion process and single-cell sequencing. But in general, we didn't see any changes in fibroblast number between the different groups. So we went in and we asked them some different questions. Again, going back to our primary goal was to identify, or at least make an attempt to identify a disease-specific fibroblast, one that we could deliver, you know, unique and novel therapies to. And if there was and is a disease-associated fibroblast, again, this may allow us to treat this disease while limiting the impact on fibroblasts that are important for normal tissue homeostatic mechanisms and wound healing. So what we then did is we actually took these almost 2,000 fibroblasts that were within this single cluster, and we hypothesized that there's an activated subpopulation central to the fibrotic process. And if you look at this cluster, you can see that, you know, the bleo, there's more red up here, and the saline are more grouped to the bottom, although they don't form distinct clusters, they do segregate a bit within that cluster. And what we did is to really to try to think independent competition to identify a unique population of fibroblasts, supervised clustering. So we just clustered, we took these 2,000 cells and we looked at how they form unique clusters and can we learn from that clustering of the fibroblasts. And we did this using genes that were known to be associated with bleomycin. We call it our bleo consensus signature. So it's a gene set that we collected in-house from bulk sequencing of genes upregulated in the fibrotic mouse lung, but also taking into account published data sets. We have a set of genes that is, again, upregulated in bleo, and we look to sort of cluster the cells based on whether they segregated on this known gene list. And then we applied a composite score called a disease score based on all of these approaches. And we hope that 3 of these different techniques applied to this population would provide some insight on what does an activated disease-relevant fibroblast look like, and how does that compare versus the healthy. And probably the most important question, is there anything absolutely unique to the activated fibroblast that we can take advantage of moving forward therapeutically? So the first approach was the unsupervised clustering. And this is where we can generate cluster the cluster. Again, the Tisney plot here, and what you can see is by taking those 1,945 cells, subclustering those out, we're able to see 8 subclusters, we call them. And each of those, again, you know, listed here, we applied the same strategy, looked for what percentage of the cells within each cluster, you know, were were derived from either the saline, the bleomycin, or the bleomycin treated with nintedanib. And I think what you can appreciate here is really the red cluster, cluster 0, is the cluster of cells that we really pull from the diseased lungs, with very few cells in that cluster healthy saline. So results, and we cluster 0 are disease relevant at the first pass that we did, kind of trying to explore again that basic question of unique fibroblasts in disease. The second approach is where we again drew a line in the sand based on our gene list of genes known to be associated with lung fibrosis, this consensus signature that we talked about. And really this clustering approach and using the highest branch point in the dendrogram broke down our two groups, or clustered the cells rather into two distinct groups. We called it a left subcluster and a right subcluster. And what we can appreciate here is that, again, there's a lot of overlap in what we're looking at. These are all fibroblasts, so lots of overlapping genes, but there are distinct patterns. And when we drew this line and said, what's in that left subcluster, or what groups, cells from which groups fell into that left subcluster, you can see in that left subcluster, which is this reddish color, the salmon color here, that left subcluster was made up of cells primarily from the bleomycin group. So really disease-associated cells. And what we found really interesting, again, based on the clinical benefit of nintedanib, nintedanib actually was able to shift cells sort of to the right, into the right subcluster. And this right subcluster was primarily derived of cells coming from saline. So almost all of the saline cells fell on this right side, whereas most of the disease cells fell on this left side. So we kind of call this left subcluster the disease-relevant population. Again, down here I can see and just show that this heat map, that extracellular matrix genes, which are all grouped down here, are very rich in that left subcluster. So that was our diseased cluster from approach number 2. And then finally, we used we generated a Z-score, a composite score for each of the cells, and we generated a waterfall plot here. And what you can appreciate is this scoring system differentiated cells based on the previous two methods. And you can see the bleomycin and the nintedanib treated cells had higher enrichment scores. So these are coming from the disease-associated tissues. And the saline fell to the left of the waterfall. We then took we did another subset of this total population, and we looked at just the bleomycin treated fibroblasts. So just cells coming from the disease. And we performed that same analysis. And you can see there's a distribution even within the disease cells. There are cells with higher Z-scores, so higher enrichment score. I don't want to say more disease, but at least a higher enrichment of the genes that we're associated with this disease state. And there are some on the low end. So what we then did is we took just the top 5%. So the cells with the most distinct, highest enriched scores and the lowest, and we looked at genes associated again with these two sort of high and low. And what you can appreciate here is the top 20 genes that were in the top 5% ends, or the bottom 5%, just pull them out here. It's genes that you'd recognize from fibrosis work. I mean, smooth muscle actin, tenacin C, latent TGF beta binding protein 2, a number of different collagens, 1A1, 1A2, collagen 5, decorin. So again, genes that are known to be associated with matrix, with wound healing, with fibrosis. And these are genes that they're not just matrix producing genes or genes involved in matrix production or turnover. But they're genes, that are involved in cell signaling, proliferation, production, turnover. So a lot of different pathways were identified here. And again, just to point out the conclusion, to make sure this point, the signature score that we had here validated the first two approaches, and we had 36 of the 38 cells with the signature score in this top end were also found in the other two methods. So these three methods apply kind of come as a consensus. Very important identified in that population using all three methods. And we wound up with 49 genes that were identified by all three methods to sort of define the activated fibroblast. So I just want to make the point, again, again, coming back to smooth muscle actin and some of these other genes that in the fibrosis world we look to as ways to distinguish an activated myofibroblast from a fibroblast. Yeah, it's on the list. But we really think that in order to truly define what an activated cell is, we may need to look beyond just single genes and obviously to a composite score, something like this. So we identified 49 genes, and just wanted to make the point in a few of the genes that I've highlighted here. So for instance, smooth muscle actin, which is the green row right here, ACTA2. This and this is fold change in sort of inactivated versus activated. So it was upregulated absolutely in all three, or utilizing all three methods, we saw upregulation. But if you look at the percent of cells expressing, so only 64% of the activated fibroblasts. So even in that activated subcluster, only about 64% of the cells express notable smooth muscle actin. And smooth muscle actin is also expressed in a number of other cell types, non-fibroblasts and non-activated cluster here. So again, if we use smooth muscle actin as a sole indicator of what a disease fibroblast is, it's going to really limit our interpretation to potentially just a subset. Spark, thymosin beta 10, a couple of other genes here that were actually expressed in 100%, including, I say, I should probably highlight collagen 181, 99.8%. So matrix genes and a few of these other matrix-associated proteins like Spark, which is used quite widely as a marker of matrix and wound healing. They're expressed in basically the entire activated fibroblast population. So again, we isolated fibroblasts, we subclustered fibroblasts, so we have fibroblasts. But if you also look at those genes, they're expressed in non-activated fibroblasts. So they don't really give us a way to distinct activated versus non-activated. Now, these genes are upregulated for sure in the activated state. Probably our best single indicator of activated population, at least based on our methods, is growth factor beta protein 2, which was expressed in about 80% of the activated cluster, only 8% of the non-activated fibroblasts, and really less than 2% of the non-fibroblasts. So this one really did give us kind of a unique gene that was associated at least with our activated cluster as we defined it. So now, one of the things that I could have made hopefully clear a couple different times throughout the presentation was the single cell sequencing has its obvious benefits. You can learn a lot of details about these different populations. For instance, this is just our fibroblasts. We had 17 other clusters, or here 16 other clusters that we can dive into and look to see how they're changed in disease. But there are some caveats, and one is the sensitivity of the system to be able to pick up genes, and we may be just losing expression of smooth muscle actin in cells, or really we may not be isolating all of the populations from the tissue, or the cells that we do isolate may not live through the process, so they may not be captured in the final analysis here. So we kind of always have to take a step back. And one thing we do in my lab, and we try to do this for as many studies as we can, is we take and we go and we do in situ hybridization. So a technique called RNA-scope here, because we want to look back to the tissue and see if the findings that we observed from the single cell approach, see if we can translate those back and at least get some insight at the tissue level, not just to the spatial distribution, but really to the expression pattern of some different genes. So Ted and my group runs this assay, and on the left here is saline, and on the right here is bleomycin treated lung, and you can see the architectural differences clearly. But we are actually doing, again, RNA scope and in situ hybridization technique for smooth muscle actin, LTBP2. As I just mentioned, this was a marker, one of our best markers for the activated population, and then collagen 181, which should be in all fibroblasts. And in the healthies, you can really see there's again a really nice pattern, I think, that mimics what you would anticipate from a healthy lung tissue, where smooth muscle actin is around the vessels, and you do see some collagen 1A1 expressing cells, the little blue dots here in the interstitial space, but very few. And you do see a few cells with LTBP2 again in the healthies, and in the non-fibroblasts we saw this in about 8%, 7 or 8%, I think, of the of the cells we isolated. Now in the bleo, it gets messy a little bit, but you can see the upregulation of smooth muscle actin with the green, LTBP2 in the red, again, consistent with what we found, and collagen 1A1. So most of these cells have blue. So the purple is sort of the combination of the red and blue. And what you can see though, as again, I kind of pointed out with the single cell sequencing, there are cells that express just smooth muscle actin. There are cells that primarily express LTBP2. And then there are cells that co-express. You can see the purple color here made of the red and the blue, which is LTBP2 and collagen. And there are also a number of triple positive cells or cells that are COL1, that are smooth muscle actin and COL1A1 positive. So when we looked at this, again, it's not always possible to make a perfect correlation between what we see in the tissue and what we were able to collect and isolate and sequence using the 10x single cell platform. But we do believe that these data really demonstrate the heterogeneity of the cells that we sequenced and the different patterns. And we can translate sort of what we were seeing, at least in the mouse on day 11. We can make good connection between what we're seeing visually in the tissue and what we observed using the single cell sequences. And I think this also, again, just to make this point again, these data also highlight the challenges of using any single marker to identify the fibrotic population in diseased tissue. Because again, if you pull out just a single marker, you can get an index of disease severity. I do believe that. But you're going to be missing probably some other populations that are really going to be important to understanding the disease process, pathogenesis, progression, and maybe identifying treatment options. So with that, I appreciate your time here and just wanted to finish up with a brief summary. This is what we did, lung cells on day 11, and we generated using 3 complementary, really a gene score rather than identifying a unique gene. I mean, really that was sort of the take home, which was, important to make, but a bit discouraging that there really was no single gene that identified the disease population. They're fibroblasts, and they do have differential expression patterns. They upregulate these matrix genes, but there wasn't a unique gene, cell surface receptor, something that we can say we can take advantage and simply deliver therapeutic, drugs in a way to just that disease relevant population. That wasn't there. But I do think this 49 gene signature can help us understand and be able to better characterize the disease associated cells. And of course, doing that at different time points throughout the model would be important. And I also think, it's important to recognize, and we focus in on one cell population, the fibroblasts here, but of course that's really just a piece of the large puzzle that occurs within the fibrotic lesion. There's going to be inflammation, ongoing tissue injury, damage. The matrix production in the fibroblasts are of course playing a massive role in the amount of matrix that's being produced and how it's being processed and laid down and cross-linked. But there are going to be many other cell types that we should also look into and see if there's ways to harness and target these cells. And one, just to make the point, was the macrophages, because these cells come in and they tend to stick around even beyond and past the initial insult, and most likely are delivering inflammatory mediators, cytokines, growth factors locally to the tissue of interest, in this case, the lung. And I do believe that that's an important population, as many do, and really a lot of great work and publications on the role of macrophages. But therapeutically, that might be a strategy that we can move forward versus maybe thinking about the fibroblast population for drug therapy. And just to thank the group, Gabor and Rebecca from Molecular Profiling, they have done and continue. Rebecca is now in grad school, but Gabor continues to lead the cardiovascular efforts, and the single cell sequencing, the analysis, and the molecular profiling efforts really were championed by Gabor. Ted, Yingling, and Lewis were doing the in vivo and the cell isolation work as well, and then a number of different individuals, Yim and Christina from our tissue core and our RNA core, actually running the 10x and delivering those data. So I think with that, yep, that's it, the last slide. So I really appreciate everyone's attention, and we'll field any questions that you have.
Thank you, Scott. That was a great presentation, and we actually have plenty of questions coming in. But for those who are have questions and haven't posted them, you should note there is a questions panel that you can type in, and I will read out the questions to Scott. So as I said, there are quite a number of questions. Let me start with the first one. You alluded to you alluded to not finding a single gene, but strategy. And this question asks about actually targeting intracellular communication. And to note, add some colors to that. Yes, we've seen similar kind of work where people look at, in terms of neural work, we're looking at ligand receptor pairs that might be upregulated or downregulated in the disease state, or so that might suggest new pathways to target. Have you considered this approach?
So yeah, so let me see if I get the question. So looking at ligand receptor. So yeah, so to the point we have, one of the things we do, and I agree. It's a great question. I agree that a lot of the regulation in fibrosis and in many different disease settings occurs very locally. We were focused here, you're right, on kind of looking at cell surface markers for the most part that might be unique to a fibroblast coming from disease versus healthy. But I do think that the local regulation of, in particular, let's say growth factors, and their activation locally within the fibrotic lesion is something that should be considered and taken advantage of. And I know there's some great work looking at, TGF-beta activation locally, and ways to sort of modulate and keep it in its latent form. And again, all that activation occurs very locally to where the fibrosis is driving disease. So yeah, that's an important point and something that I do see the field moving toward, and I think it's the right direction to be considering here.
Okay, thank you. Well, this going back to earlier in your presentation. This questioner asks about the difference between cluster 2 and cluster 4 from the IPF patients. Do you have any information on the locations of the fibroblasts? And do you know which one might be more sensitive to pirfenidone?
Oh, that's a really great question. No, it's really interesting. I think the in situ hybridization, the RNA scope, and now techniques looking at spatial transcriptomics coming also from 10x. We're starting to apply those, but we haven't done that systematically. Where we would really like to be able to, and it's a great question and point, because we'd really like to be able to not only, let's say if we had human tissue, perform the cell isolations, run the 10x, look for distinct populations, and hopefully finding maybe more of a smooth muscle actin contractile population versus a matrix rich population like clusters 2 and 4, but then also go back to the tissue. And using the spatial transcriptomic approach, you can actually do gene expression at this single cell resolution level in the tissue. And it's based on a grid and a pattern, and then you can go back and look to see not only what cell types may be expressing what different genes, but you can look to see where they're expressed and their co-localization to maybe a macrophage or to each other. So yeah, so being able to find where these cells are and their differential response to different drugs like pirfenidone is really a great question, and one that I think still needs to, I think the technology is there now, and we're working on it. I know the field in general is working on that question.
Thank you. The next questioner asks, what would be your approach to target the bleoactivated fibroblast population when it seems to be that there is sort of a gradual changes in expression?
Yeah, there's a
different
gradient.
cell types.
Yeah, that's an awesome question and really an important one. When we look at these waterfall plots, it's not black and white, and nothing here is. These are all fibroblasts. and there is a gradient in expression. Again, there's no unique gene that sort of fell into the activated versus the saline or the healthy. It's one where for instance, looking at the expression, so there may not be a unique cell surface protein in the disease versus the healthy, but looking at the overall pattern. So for example, and again, this didn't fall out into our 49 gene list, but looking at something like PDGF receptor alpha, this is a receptor that will internalize quite readily. It tends to be expressed primarily in fibroblasts. It's upregulated in many fibrotic diseases. So it may be a way to really take advantage. Again, you're not going to be limiting, a treatment if you use something like a PDGF receptor to the disease population, but you may be able to enrich, your drug into that cell population by using a receptor like that. So I do think you have to look across that sort of waterfall plot, and you're right, nothing's unique there. There's just different levels of expression, and if we can take advantage of the ones on the high end to concentrate therapies to those cells, I think that's a worthwhile approach as well.
Thank you. This is somewhat related. Another question related. If you had a model for macrophage activity in the lung activity, how would you integrate the variety of their gene expression into a cell-based model?
It's a good question. Not one that I've thought about. So moving it into a cell-based system, you're right. It's again, the macrophage population, it's the infiltrating cells. So I mean, we would think to first, it's really trying to be able to subcluster that macrophage population and distinguish interstitial versus alveolar. Again, one sort of being more disease relevant and the other being, critical, to normal homeostatic mechanisms within the lung. So maybe focusing then on the interstitial and using those and trying to identify if there are any distinct gene expression or cell surface markers from those. But when we do look at that macrophage population in more depth, so we do subcluster and you apply, a number of different macrophage markers, CD68, CD64, CSF1R. I mean, there's a lot of MSR1. There's a lot of really interesting markers, but they tend to highlight fairly large percent of the macrophages and not necessarily those that are disease associated. So we'd apply almost the same filtering approach to find what's the relevant disease associated macrophage, and then again try to find ways that we can deliver therapies just to those without impacting cells elsewhere in the body. If you can identify the right cell surface marker to highlight that disease associated macrophage, we may be able then to do an isolation and put those cells into culture and sort of use that as a culture-based system, but it's going to be challenging.
Thank you. So we have a few more questions, and if it's okay with you, Scott, I'm gonna let this run over the hour because we've reached the well, it's 1 o'clock on East Coast time. But so here a technical question in terms of do you think there's value in comparing the outcome of single cell RNA-seq versus nuke-seq?
Absolutely. That's an awesome question. I appreciate that one. We're actively doing that. And I know, again, it's not that's a novel approach, but I do think that, in the literature, people use one or the other, but I think they both have benefit. Single nuclear sequencing for us, the way we look at it is it's a unique opportunity to go into tissues and biobanks that have been frozen down. Single cell sequencing, as the way we use, we're using fresh tissue. And that gives you obviously the best approach to get viable cells through the 10x process. But single nuclear sequencing, I do think there'll be caveats there. But I think it's an important question to do a head-to-head from the same animals, the same tissue, the same disease state. Really see an analysis of what you pick up with cell over single nuke and the reverse. But it's an area I know that we would like to move, and we're starting to do some pilot work because I really want to be able to get my hands on human lungs, and many of the biobanks that have tissues, they're sitting in -80, and they're not going to be viable for single cell. but they would for single nuke.
Right, right. Thank you. So could you comment on, in terms of the 49 gene signature, whether or not this is conserved in other fibrotic models or even in cancer?
Awesome question. Let me pull up. Primarily, yes. I think the different patterns, and this is an area, as I showed that initial image of fibrosis across organ systems. It's really something I think I personally need to do a bit more and spend a bit more time with, and as a field we do as well, is looking at these different gene data sets that are available in the public domain, and really trying to put them on a platform where we can compare and contrast. For the most part, though, these genes as really matrix, osteopontin, WISP, these are genes that are really well known and associated with the matrix production and wound healing, and we do see them up in cancers and across that liver, kidney, and cardiac fibrosis models. But the systematic sort of layering of these, I do think actually needs to be done in a way that is fair. I think from the molecular profiling, from the the technical side, it's just kind of making sure that all the platforms are aligned so that the gene expression can be normalized in a way that makes sense.
Thank you. So actually, a follow-up question, a different vein from the same questioner is, can you comment on reprogramming versus killing CAFs with these antibody drug conjugates? And what's your thoughts on this?
Yeah, I think as of right now, both are viable approaches to consider. Of course, proof of concept preclinically. Reprogramming, any cell is going to be the preferred approach, I would think, only because this is going to allow, again, we want to take a disease-associated fibroblast and we want to turn it down. And if you can find the right approach to do that, of course, this is going to be a much safer approach. I do think for proof of concept, and I am a fan of sort of maybe trying the more blunt approaches, where if you can find the right marker and you can specifically deplete the cell of interest, whatever that is, that would give you, some definitive data to support its role in the process and also the safety. So from my perspective, we're looking to try both strategies and techniques to, immunotolerize or suppress an activated CAF, or in essence, get rid of that cell, deplete it, and see the outcomes there.
Right. Thank you. So the another question is whether or not you've actually looked at gene expression profiles from human fibrotic lung versus healthy.
We have not at the single cell level to date, and that was again going back to the great question around nuclear sequencing and being able to work with tissues that are banked, to get at healthy and diseased. But at the bulk level, so like many, we've looked at a lot of different bulk samples, but we do want to now move to single cell from human healthy and diseased, and then also be able to layer in, if tissue is available, the spatial transcriptomic approach, so we can look at spatial distribution of these cells.
Thanks. And actually, this is one area where we could help, of course, because we have access to the human lung, both in the form of PCLS, but also preserved lung tissue. So that's one thing that we're quite interested as well. So we're getting to. Oh, sorry, you had a comment.
Perfect.
Okay.
No, I think that's perfect. Yeah, I agree. I think the lung slices is a great.
I think we got cut off a little bit, Scott, but that's okay. We're now getting to 5 after 1, and I want to actually thank both. I want to really thank you, Scott, for a great presentation, and that we really enjoy working with you in terms of collaborating, and we appreciate you coming on to give this talk. And I also want to thank all those attendees who have joined and asked, listened to the talk and asked so many great questions. So if you can, if you want to listen to the talk later, the talk is being recorded and will be available at our website. And please join us for future webinars. We are doing this practice on basically on a monthly basis. So I want to, again, I thank you again for joining us, and have a great day and have a great weekend.